Hello everyone. Today I want to talk about something that sits at a genuinely uncomfortable intersection, a nutrient that is critically important, almost universally underconsumed in modern populations, and increasingly being supplemented at doses that the research is beginning to flag as a source of the very problems it is supposed to prevent: vitamin D, and specifically, the pattern of aggressive high-dose supplementation that has become normalized in the wellness space, driven by widespread deficiency awareness, enthusiastic supplementation advocacy, and a simplistic framework that treats more as categorically better without accounting for what the gut microbiome does with the excess. I want to start by being clear about what vitamin D deficiency actually is and why the concern is legitimate. Vitamin D insufficiency is genuinely prevalent. Conservative estimates place over 1 billion people globally in the deficient or insufficient range. With young adults living in urban environments, working indoors, and spending limited time in natural sunlight among the most affected populations. Let me start with how vitamin D actually interacts with the gut microbiome, because this is the biological foundation that makes the overcorrection problem real. Vitamin D receptors are expressed throughout the gastrointestinal tract in intestinal epithelial cells, in immune cells of the gut-associated lymphoid tissue, and in the enteroendocrine cells that regulate digestive hormone secretion. Vitamin D signaling through these receptors plays a direct role in maintaining tight junction integrity, calibrating the gut immune response, and influencing the composition of the microbial ecosystem. This is why vitamin D sufficiency is associated with better gut barrier function and more favorable microbiome profiles. But the vitamin D receptor pathway is dose-sensitive. It is a nuclear receptor that modulates gene expression, and like any gene expression pathway, it operates within a range. Below that range, function is impaired. Within the range, function is optimized. Above the range, the same pathway that supports gut barrier integrity and immune calibration can produce outcomes that run in the opposite direction. The second layer of the problem is the calcium-gut interaction, and this one is rarely discussed outside of nephrology contexts but is directly relevant to anyone taking high-dose vitamin D without careful monitoring. Vitamin D's primary classical function is the regulation of calcium absorption from the gut. At physiological levels, this function is tightly controlled and essential. At supraphysiological levels, vitamin D drives calcium absorption beyond the body's capacity to manage the excess, producing a state of relative hypercalcemia, elevated calcium in the bloodstream that has multiple downstream consequences. The third layer is the vitamin K2 interaction, and this is the one that produces the most clinically significant consequences and is almost universally absent from conversations about vitamin D supplementation. Vitamins D and K2 work together in a metabolic partnership. Vitamin D increases the production of several calcium-binding proteins, including osteocalcin and matrix GLA protein, that are responsible for directing calcium to bone and preventing its inappropriate deposition in soft tissues, blood vessels, and the gut wall. These proteins require vitamin K2 to become activated. Without adequate K2, the calcium that vitamin D mobilizes has no reliable guidance system. It ends up in places it should not be. The fourth layer is the magnesium dependency, which compounds the K2 problem and creates the conditions for a supplementation cascade that the gut pays for. Vitamin D is fat-soluble and requires enzymatic conversion to its active form, first in the liver, then in the kidneys, through hydroxylation reactions that are magnesium dependent. When you supplement with vitamin D in the context of magnesium insufficiency, which, as I have covered before, is extremely prevalent in modern diets, the conversion pathway is impaired. The supplemental vitamin D accumulates in its inactive form. Serum levels may appear adequate on standard testing, and the cellular availability of active vitamin D remains low. This leads people to increase their dose further, compounding the magnesium depletion as the conversion attempts consume what little magnesium is available. So what should you actually do with all of this? If you are vitamin D deficient, genuinely deficient, with a serum level below 50 nanomoles per liter, correcting that deficiency is worth prioritizing. But the correction should be guided by testing rather than by generic dosing recommendations, and it should account for the cofactors. The correction depends on magnesium sufficiency should be established before beginning supplementation. Vitamin K2, specifically in the MK7 form, which has the longest half-life and most favorable tissue distribution, should be supplemented alongside vitamin D at meaningful doses, not in the negligible amounts included as an afterthought in some combination products. Supplementation should aim for sufficiency. Serum levels in the 75 to 100 nanomole per liter range, not for the supraphysiological levels that high-dose protocols produce. and testing should be repeated at intervals to ensure the correction has not overshot into the range where the microbiome disruption and calcium management problems begin. According to research, the gut microbiome benefits of vitamin D are concentrated in the correction of genuine deficiency, not in the continued elevation of levels beyond physiological sufficiency. The dose-response curve is not linear, it curves, and on the far side of optimal, the direction of the curve reverses. More is not better. Sufficient is better. And sufficient, for most people, is considerably lower than what the wellness industry's vitamin D enthusiasm has normalized. I hope this gave you a more nuanced picture than the standard deficiency narrative provides. Drop a comment and tell me what dose you are currently taking and whether you have had your levels tested. I genuinely want to understand how widespread the overcorrection pattern is. If this was useful, hit the like button and share it with someone whose supplement stack includes a high-dose vitamin D with no K2 and no magnesium. And if you are new to Nutriflow, subscribe and hit the bell. See you in the next one.